Continuing on in our exploration of the nervous system, we begin our part 3 here. We left off just about talking about the synapses and the different types of connections that we have. So a synapse itself, we have essentially two components to it. We have, when we're talking about two neurons that are connecting together, we have a presynaptic neuron and we have a postsynaptic neuron. And so the action potential is traveling along the presynaptic neuron towards that synapse, and the action potential then moves away from that synapse in the postsynaptic neuron that is there. But the postsynaptic cell could also be a muscle cell, a gland. It doesn't always have to be a neuron that we're talking about. And so we talked about last time that we have different types of synapses themselves. And so we have everything from axosomatic types of synapses, axodendritic, axonic types of synapses. Essentially anywhere where that axon can make a connection, we can potentially have a synapse that's there. And so axodendritic, obviously between the axon and the dendrite, axosomatic between the axon and the cell body, the soma. And then some other less common ones, we have things like axoaxonic communication, dendrodendritic, and dendrosomatic. When those cells communicate with one another, we have essentially two ways to convey that information across the synapse. We either have an electrical synapse or we have a chemical synapse. Electrical synapses are more rare. We typically only see them in the CNS itself, and this is where we're going to have that electrical potential jump, in a sense, directly from the presynaptic neuron directly to the postsynaptic neuron. So ions are going to be conveyed directly to each other. This type of synapse is the equivalent of like wiring two wires together. Putting the two wires together, the electricity basically goes directly from one wire to the next. A chemical synapse utilizes neurotransmitters to span that gap. Those neurotransmitters have to go into the synaptic cleft, the space in between the presynaptic and the postsynaptic neuron, and that's going to take a little bit of time. So we're going to have a little time delay there, and it's going to have to bind to that postsynaptic neuron or to the postsynaptic cell, the effector cell. And so this process is utilized the majority of the time, and we have a number of different chemicals that we can use as neurotransmitters to transmit this information from one to the next. So that synaptic cleft is just the space in between the two cells that we have connected to one another. It is a fluid-filled space. In a chemical synapse, we don't have a specific bind or bond between the two cells. They're not directly connected to one another. So we're going to have a little bit of a space there with fluid, extracellular fluid that fills that space. That's going to allow for that neurotransmitter to cross across the synapse itself. That's there. And so this is a chemical process. We're going to have our neurotransmitter move across the synapse, bind to a receptor, and then do something to that postsynaptic cell. That movement of that neurotransmitter is, in a sense, the transfer of information. And so this is where that neurotransmitter is allowing for the movement of that action potential, the signal that something has happened or something needs to happen from one cell to the next. And when that action potential is coming down the presynaptic neuron, as it starts to make its way down through there, it opens up voltage-gated calcium channels. Those voltage-gated calcium channels allow for calcium to migrate into the axon terminals, and the axon terminals then have our neurotransmitter vesicles containing the neurotransmitter itself. And that influx of calcium is going to trigger the opening of that neurotransmitter into the synapse. And so we're going to exocytose the neurotransmitter out of the presynaptic neuron into the synaptic cleft. It's then going to migrate across, bind to a receptor on the postsynaptic neuron, and then from there it's going to make some sort of change in that postsynaptic cell. That postsynaptic cell may immediately open up ion channels. It may close ion channels. It may cause the cell to start to manufacture something. It all just depends upon what that neurotransmitter and receptor combination leads to. So here we have that neurotransmitter process in the synapse itself. And so calcium is going to come into the inside of our presynaptic neuron. That's going to cause the migration of vesicles towards the axon terminals. And those vesicles are then going to exocytose our neurotransmitter to where it then binds to a receptor. And in this particular case, it's going to cause the opening of a sodium channel, and sodium can then rush in to the inside of the cell. So this postsynaptic neuron here could potentially be back like when we were talking about muscle. That neurotransmitter is then going to be broken down by a enzyme. And so when we talked about muscle, we talked about acetylcholine being our neurotransmitter and acetylcholinesterase being the enzyme. So acetylcholinesterase can break down that neurotransmitter, and when that neurotransmitter then leaves the receptor, that then causes the receptor channel to go back to its resting state. In order for us to stop that postsynaptic neuron from, in a sense, continuing to have a signal and continuing to do whatever it's been told to do, we need to end that signal. And so we have to terminate the neurotransmitter's effect on the postsynaptic cell. And so that neurotransmitter that is there is going to have essentially a continuous postsynaptic effect as long as it's bound there. So we need to be able to get rid of it either by using enzymes, like we just talked about. Some of that neurotransmitter that gets out into the cleft doesn't necessarily have a chance to bind to the receptor itself because it diffuses out of the synaptic cleft itself. And then from there, some of it may be reabsorbed by things like astrocytes or directly into the presynaptic neuron. And so that then eliminates our neurotransmitter from the synapse, and we can move on to the next signal. That movement of the neurotransmitter from the presynaptic neuron to the postsynaptic cell creates a little bit of a delay. And so we have this synaptic delay that occurs in the transmission of nervous information. And so depending upon the particular cells involved, this can be several milliseconds in terms of the length of time that it takes to get across. It doesn't sound like a lot, but in the grand scheme of things of nervous transmission, this can be, in a sense, a lifetime. And so that synaptic delay is the one thing that really kind of limits how quickly we can send information through the system itself. And so that's where we get this synaptic delay from, and it's going to slow down every time that we need to make a connection from one to the next. When that presynaptic neuron releases its neurotransmitter or moves the electrical charge across to the postsynaptic cell, what happens in that next cell is not necessarily always guaranteed per se. And so it's not guaranteed that we're going to cause something to happen in that postsynaptic cell, because we can have excitatory potentials, and then we can also have inhibitory potentials. And so not only does factors that are impacting the synapse itself play a role, and so how much neurotransmitter was released, the amount that is there, how long does it take, and how long can those neurotransmitters be actually bound to the receptors, how quickly are they broken down in the synapse, all plays a role. We talked about a bit about this in muscle when we talked about things like Botox affecting the ability of muscle to perform. So EPSPs, or excitatory postsynaptic potentials, are going to allow for us to essentially start to depolarize that postsynaptic neuron. And so these are excitatory postsynaptic potentials. These are potentials that are causing the postsynaptic cell to start to depolarize. And so they're graded potentials. IPSPs are going to cause the cell to start to hyperpolarize. And so this is going to make it so that they become more and more negative, versus the EPSP makes them more positive in terms of the charge on the surface of the cell. And so an EPSP, if our normal resting membrane potential is at negative 70 millivolts, an EPSP is going to cause a little bit of a blip up towards threshold. But it's a graded potential, so it doesn't get us there. An IPSP It's going to take us from that negative 70 millivolts and bring us down further into the negative. And so that means that even if another stimulus came along in this point here, we would be hyperpolarized and it would be a much more difficult process to get an action potential to occur. Any given postsynaptic neuron is going to have potentially a large number of presynaptic cells that are actually firing upon it. And some of them are going to be giving off EPSPs, some of them are going to be giving off IPSPs. And so with that, we're going to have to look into summation that is occurring within the postsynaptic cell. And so we can have temporal summation. This is where the action potentials from the presynaptic neuron are just coming very quickly. And so they're sending signals very, very fast. Spatial summation is going to be where we have the presynaptic neurons firing off at the same time, but they're all concentrated into a small area. And so they're all into a small little area. So we have a spatial type of a summation that's going on. And so we're going to add up all of the EPSPs, all of the IPSPs, and see what ends up happening in that postsynaptic neuron. And so here we can have our EPSPs. And so here we have an EPSP separated by time, nothing happens. It's not close enough to the first one in order to get an action potential to occur. But if we shorten the distance in between excitations, now those two EPSPs add up to enough to get us to threshold. And so now we're at that threshold point. Now we're going to get our action potential, our depolarization, and we go on from there. If the both of them fire off in the same proximity at the same time, they again can add up to one another, and that's going to lead to our action potential having occurred in the postsynaptic neuron. IPSPs generate a hyperpolarization, and so it's going to drop us further into the negative as far as the charge goes. But altogether, we've got to add up all of the EPSPs, all of the IPSPs, so we can have other neurons coming in here giving us IPSPs. We can have other neurons coming in here giving us EPSPs. And depending upon which one, in a sense, is winning, if in the example here, if the IPSPs and the EPSPs just simply cancel each other out, we don't really see anything. Maybe we see a little bit of a depolarization, maybe we see a little bit of a hyperpolarization. It just depends upon the total summative component of all of those different presynaptic neurons. In order to communicate from the presynaptic neuron to that postsynaptic cell, we need to have a neurotransmitter for the most part. And so neurotransmitters are just a protein that is being produced by the neurons themselves. And so we've got some 50 plus different kinds of neurotransmitters that have been studied. The most studied one is acetylcholine. And so acetylcholine is going to have the most study on them. It was first discovered on muscles, and so we have the ability to stimulate skeletal muscle very easily. It's easy to study, it's easy to work with, and so on. And so this allows us to get quite a bit of information about it. For the rest of the neurotransmitters, I want you to know the general class of the neurotransmitter and what it does. And so what's the general class? So biogenic amines, these are things like emotions and biological clock. Amino acids are for communication within the central nervous system. So we use them primarily there in the central nervous system itself. We have peptides. And peptides are kind of our feel-good neurotransmitters. So these ones are going to be the ones that take away pain when we need them to. And so we can do things even though we're in pain. We may not necessarily even notice that we've hurt ourselves at times, because all of that neurotransmitter is blocking the transmission of pain at that point within the central nervous system itself. And then we have what we call novel transmitters. And so these novel messengers that we have are substances typically that we don't necessarily associate with being a neurotransmitter. And so things like ATP, as well as gases like nitric oxide and carbon monoxide. And so they all can function in a neurotransmitter function as well. The neurotransmitters themselves can also be either excitatory or inhibitory. And so that excitation and inhibition is going to typically depend upon the receptor that we're using. And so the neurotransmitter can have an effect depending upon the type of neurotransmitter receptor that we have, and can then make it either an excitatory or an inhibitory type of substance that is there. And so different types have different effects on either depolarization or hyperpolarization of the particular cells. But in the end, it's the receptor that is the important aspect, because we can have the same neurotransmitter, in this case acetylcholine, work as both an excitatory and an inhibitory type of a neurotransmitter. So we talked about it in skeletal muscle, and in skeletal muscle it is excitatory. It caused muscle contraction. But acetylcholine in cardiac muscle is inhibitory, and so it slows down the heart contraction. So it all comes down to that receptor and what it's going to do for that postsynaptic cell. When that neurotransmitter binds to the postsynaptic cell, it can have an immediate effect. These are direct neurotransmitters. This is what we saw with things like skeletal muscle. And so acetylcholine works directly on skeletal muscle to open up ion channels. And so it's going to immediately open them up. Other neurotransmitters work indirectly. And so they're going to have a little bit longer of a process in order to get their effect occurring in the postsynaptic cell. They do so by using what are called second messengers. And so the neurotransmitter is the first messenger, and the chemical that is produced inside the postsynaptic cell is our second messenger. So in our direct acting cells, and so when we have a neurotransmitter that is direct acting, that neurotransmitter binds to the surface of the receptor, and it either opens the receptor, potentially closes the receptor. It may turn on a process in the cell. Whatever it happens to be, it's going to happen immediately, and it's going to be relatively short-lived. And so it's going to not last very long when it occurs there. And so we can get depolarizations, we can get hyperpolarizations, depending upon the receptor that is there. G protein linked receptors are our indirect pathways. And so indirect receptors are going to typically use a G protein linked type of receptor. These are going to be a little bit slower. They're going to give us a little bit longer lasting effect in the target tissue that we're working with there. And so a G protein linked receptor has a neurotransmitter that binds to a receptor on the outside of the cell. And so we have our little receptor that is there, but this receptor doesn't have a channel going through it. So when our neurotransmitter binds to it, it doesn't open anything up inside of it. It's going to initiate a series of chemical reactions. And those series of chemical reactions is going to begin with that G protein activating GTP and hydrolyzing it into GDP. So we're going to take off a phosphate group, similar to breaking down ATP. Now that our G protein has been activated, we can go through a series of processes that activates adenylate cyclase. That adenylate cyclase then serves as another enzyme which catalyzes the formation of cyclic AMP. And so we get AMP from ATP. So this phosphorylates the ATP. We get that breaking down through hydrolysis, and we end up with cyclic AMP. Cyclic AMP then can be our second messenger. This is now the second messenger that's going to go out into the cell, and it's going to tell the cell, in a sense, what it needs to do. And so this is now inside the cell. This neurotransmitter never entered the cell. Here's our plasma membrane there. It never entered the cell. It never had anything affect the inside of the cell other than to start this cascade of chemical reactions, which gets us down to our cyclic AMP, and that cyclic AMP now becomes our second messenger. That second messenger inside there can then activate protein kinases. Those protein kinases are enzyme activation. Can then go and start some other processes inside the cells. It may turn on some specific genes. It may open ion channels. It may close ion channels. Whatever it happens to be, it's doing it inside the cell now. Cyclic AMP is one of the major second messengers that we have, but we have many other second messengers as well. Cyclic GMP, diacylglycerol are all different types of second messengers that can be used inside the cells themselves. The chemical process is just going to be slightly different depending upon which one we're using. And again, that second messenger makes a change inside that postsynaptic neuron. Something is going to occur inside that postsynaptic neuron, turn something on, turn something off. All depends upon what that neurotransmitter and receptor gets us. When neurons communicate, they're going to communicate through kind of oftentimes groups of neurons. So if we take a pin and touch the tip of our finger, we're not just touching a single neuron, we're touching a series of neurons that are going to receive that information. And then that information is going to go out from there. And so in a neuron pool, in a simple type of pool, we have essentially an input fiber. And so that input fiber comes in. This is kind of the central portion there. We then have an area where we have a discharge zone. And then we have an area that is a facilitated zone. And so if that one neuron was coming in, the discharge zone is the area closest to that neuron as it came into the pool. And then the further ones in the facilitated zone are a little bit further away from the original stimulus. That information as it comes in, especially to the central nervous system, has a couple of different potential pathways that it can follow. It can be divergent, meaning that we take a single piece of information and we send that information out and kind of broaden the horizon in a sense. And so this oftentimes amplifies what the original stimulus was. They can all end up going in basically the same path and get to the same place, or they can end up going to different places. And so they can be sent to different areas of the central nervous system itself. Convergent types of pools rely upon taking multiple pieces of information and converging it down into one place. And so we have a single output as that information comes in. And so this can be kind of like, in a sense, taking the information on a test, reading the question, taking all that information in, and coming up with a single output. We can also have the input coming from a single neuron, but that one neuron has multiple synapses on that postsynaptic neuron. Reverberating pools basically feed back on itself. And so reverberating pools are going to be kind of like a positive feedback loop in a sense. We have to get rid of that initial stimulus in order for a reverberating neuron to turn off, and to turn off that pool itself. We're going to have parallel after discharge. And so here we have, again, a single piece of information in a sense that comes in. It sends its information out to a whole bunch of different places. And then we have a single output, a single answer to the question itself in a sense. So serial processing is where everything is going along the same pathway. And so it's going to a specific spot in the end. And so we saw many different varieties of serial processing that is occurring. Serial processing is what we see when we look at things like spinal reflexes. And so spinal reflexes are kind of an all or nothing type of an event. It either happens or it doesn't. We have to have enough stimulus in the original cell in order to get us to that point where it's going to fire off into the process. We can do parallel processing. And so parallel processing is where, again, we have that single input that comes in, but it goes out and it diverges. It goes out to many different areas. And so instead of diverging towards the same goal, they go out in multiple areas. And this is where things like a smell or a sound can bring up a lot of different memories about it. You hear a song that was in your high school years, something like that, and it reminds you of kind of where you were and what you were doing and things like that while you were listening to that particular song, or even an odor, things like that as well. When neurons develop, their development itself is occurring within the primitive nervous system, and we have what we call neural tubes, and we have the neural crest. And so the neural tube is going to become the central nervous system, and this is an important aspect for development itself. That neural tube is going to eventually start to proliferate cells that are either going to migrate towards the cephalad portion, the head, or towards the caudal portion, the tail. And so we're going to start to make the primitive nervous system out of that neural tube that's there. This is important for development because this is all happening within the first weeks of development itself. And so having everything in line as far as nutrients goes and things like that for the mom is important for the development of the nervous system of the developing embryo. As those neurons continue to grow, they're going to continue to span out. They're going to be helped by things like astrocytes, and so they're going to help guide them into where they're supposed to go. The developing embryo is kind of like a big chemical soup in a sense. There's all kinds of different molecules that are floating around, and when it comes to axons, some of them are repelling them, some of them are attracting them to different tissues so that they end up growing towards the correct area within the body itself. And so we get that component of growth as it sits there. When we start to talk about the central nervous system, we'll dive further into the development of the neural tube and the neural crest cells.